All right, good morning, everyone. To those who have joined already, we're just going to wait a couple of minutes for participants to join, and then we'll get started. All right, good morning, everybody, and welcome to this webinar where we'll be talking about the data management chapter of the NSCF manual. I apologize for the video not having any sound, the intro video that we normally have. I accidentally forgot to share my sound, my computer sound before I shared this morning, but hopefully that's acceptable. So just some guidelines before we get started this morning. Please, if you've joined us and you haven't done so already, just put in the chat box, say hi, and let us know which institution you're from. And then please, if you want to ask questions, there is a question and answer feature in Zoom. If you look down at the bottom of your screen, you'll see it says Q&A. And please put your questions into that in there. If you put them in the chat, we might miss it, but hopefully with Chanel and Fulu helping behind the scenes, we'll be able to catch any questions that people ask. So this is the first webinar of the year, and I'm very happy that the webinar is on data standards and workflows. Like I said, this is related to the first chapter of the manual, and a good way to get the year started off. So just as a quick overview, we're going to be looking at the purpose of the data chapter of the manual to start. We'll then talk a little bit about data policy and data sharing. We'll look at concerns around digitization and data sharing, some important things there. We'll talk about data attribution a little bit. And then we'll get stuck into the core of what the chapter is all about, starting with data quality, looking at Darwin Core and data formats, recording data in the field, geo-referencing tools, and ending off looking at processes and workflows, which is one of the key components of work around this particular chapter. Okay, so firstly, the data chapter, it's chapter 4 of the manual. It starts on page 60 for those of you who have a copy of the manual, but it doesn't have a chapter heading. I'm very sad that this particular chapter didn't make it into the manual with a chapter heading. We did have some issues with production of the manual to begin with the people that produced it for us, and there were a couple of little glitches. And this was one of them. So there's no actual chapter heading, but it starts on page 60 with point 4.1 over there, which is a summary of the chapter as a whole. It's quite an extensive chapter of the manual. There's a lot of information in there. It's quite dense, and so we decided to start off with this sort of executive summary at the beginning of all of the important points. Okay, so why do we have a data chapter in the manual? So data are one of the really important assets that museum and herbarium institutions have and take care of. It's one of the sort of main focus areas of developing the NSCF is to try and improve the way that data are being curated and especially being published and made openly accessible. There's this global trend towards people providing their data online. So what I've got there at the bottom of the screen is an image. This is the standard Gbif Map. For those of you who know about Gbif, the Global Biodiversity Information Facility, which is a big global data aggregator. It aggregates data from museums and herbaria, as well as a wide range of other sources into this big searchable online database, and their homepage has this heat map of the density of specimen records that they've got for around the world. You can see lots of records for Europe, lots of records for North America, It's nice to see that South Africa is quite red as well. So we do have a lot of records here. And so it's that we've got this heat map of records from all over the world. And what we're trying to do with NSCF institutions is get them to contribute their records to these public platforms as well, so that the data can be used by a wide range of users for a whole lot of diverse purposes, particularly important ones like addressing global climate change and that kind of thing. And so if we're all going to be sharing our data like this, data quality and standards are important. And one of the really important things that we're wanting to get up and running as part of the NSCF project is a living atlas data portal. We've been talking about this, calling it the virtual museum, and it's basically intended to be a data portal for South African collections institutions to share their data through. It'll be like our own homegrown project where we can all, where all of the institutions can share their data. So I'm just going to step out of the presentation quickly to show you what this actually looks like. Okay, so the Living Atlas project was basically started in Australia when they developed what they called the Atlas of Living Australia. And it's basically a website that you can access to get specimen records for a whole range of different taxa in Australia. So I'm just going to show you quickly how it works. They've currently got about 102 million specimen records, which are coming in from about 10,000 data sets. So it's quite substantial. My interest is in scorpions. So if we take a quick look at scorpion, at a particular Australian scorpion genus, we can search for that genus and we get a list of possible hits. The first one is for that genus, and so I can go in and have a look at that. And it brings up a page like this, which includes a summary of information. So we've got the classification information up at the top here. We've got some images. It pulls in images from various resources. But the nice thing is this occurrence map. So it shows you a map of all of the specimen records. These are all of the geo-referenced records, of course, from a whole range of different museums and other data sources for Australia. So 10,000 specimen records. The really important thing about this is that it's aggregating data from multiple different sources into one data portal. So people wanting to get information on a particular group of organisms, like I've done for scorpions here, don't have to go and request those data from multiple different institutions directly. They can access the data directly themselves and get all of the data in one place, essentially. So, so we have the map there of all of those 10,000 records. We can also, there's various there's more details about various aspects of this particular set of data. But if I look at the data partners, this is important because it's showing the sources of where those records are coming from. So we have Western Australian Museum, South Australian Museum. It's pulling records from iNaturalist as well. We have the Australian Museum, Museums Victoria, the Australian National Insect Collection. So you're seeing exactly where the records are coming from and how many records are there, which is important. That's the attribution stuff that we're going to be talking about later. You can also drill down into that data and see more, get more specific details. So if I click on View Records and just give it a moment just to open there. So here it opens up options for being able to filter things in a bit more detail. So if I select, did this earlier, I'm not sure, and I've forgotten since I did it. There was an option here to search for, oh, here it is, okay. So, so here I might want to go and see particular species records. So I'm going to click on this particular species over here. I'm going to say refine my search and just give that a second. And then it pops up again and shows me the map of where this particular species occurs. It's down there, north of Perth. And I've now just learned something about this particular species geographic range, which I didn't know before. So we want to produce the same thing for South Africa. Basically a dedicated data portal with all of our museum and herbarium specimen data being provided openly for people to be able to use. So, and like I say, in order to do that, we need to have standards. The data chapter starts off talking about data policy, and this is really important. If we're wanting to reach the point of sharing data, we need to have policy. We would like the institutions to have a policy of open data access and open data sharing for the institution. A policy is an overarching, it's sort of a high level document that describes principles that the institution adheres to. We see quite a lot when people are developing policies that they get the policy and the implementation of the policy a little bit mixed up. But policies are essentially just these high-level documents that describes the principles that an institution adheres to. And it can be things like, we have a policy of open data access or open data sharing. Open data access is something that's becoming more and more commonplace across institutions in different parts of the world. And very often when you get funding to run a particular research project or something like that, the funders will have a requirement that the data be made publicly accessible. And for our publicly funded institutions, such as the museums and the herbaria, we believe that it's important that they have open data access policies because it's essentially taxpayers' money that's keeping those institutions running, and we want to get the maximum benefit that we can from the resources that those institutions have. So what sort of benefits are there from open data access? Well, there are a huge number. You get more exposure for your institution. People are able to use the data in ways that that data might not have been used if it was only being used internally. There are a whole range of different benefits. And but I think the important point here is that we're actually seeing this happening with institutions that are making their data openly accessible and publishing their data to Gbif. So if you go to Gbif, you can actually see the number of hits that your data get on a daily basis, and they are significant. So most institutions are getting, you know, sort of 4 or 5 or 6 or maybe even 10 hits per day, 10 data requests from Gbif per day that include records from some of our institutions. And so it really is maximizing the way that those institutions' data are being used. And it also, that can bring people back to your institution. So they might see that you have records for particular taxa in your collection, and then they get in touch with the institution and say, you know, we'd like more information, we'd like to get images of the specimens, or, you know, perhaps do you have specimens that haven't been digitized, those kinds of things. So it really does increase the use of those collections and the associated data. But there are some concerns around data as well. And sorry, is my sound still giving a problem? I see, Cameron, you said something here.
Yeah, it is still a little bit choppy.
All right, I'm going to unplug my headphones. Let's try that. Okay, can you hear me now? It's now just on the computer microphone.
Yeah, let's see if that works.
Okay. All right, so there are some concerns around digitization and data sharing as well. So one of the concerns that people frequently raise is issues around data quality. So they're concerned that there are data quality issues, that their data are not perfectly clean and perfectly up to standard, and so they don't want to share the data. It's almost like they don't want to put data out there that might reflect poorly on them or on their institution. However, the reality is that that is the case for most collections and most institutions around the world. And instead of withholding data on those grounds, it's better to publish the data as they are. You can include statements about the data. You can include qualifiers about the quality of the data when you publish the data, to say, things like, we haven't cleaned the data, we haven't got the taxonomy up to date, there might be mistakes. And then it's up to the users of the data to interpret that in terms of their particular use, how they're going to use the data. They might be able to say, well, the data is good enough for my particular application anyway, or maybe they can do a little bit of data cleaning to improve the data to meet their requirements. But the idea is we get more benefits from putting the data out there, even if the data are not perfect, than we do from holding on to them and hoping that we'll be able to improve the quality at a later stage. Another concern that people raise is about misapplication of the data. So they say, well, someone's going to take my museum data and they're going to use the data inappropriately because they don't know the details of how the data were collected, or particular, there might be biases in the data set or something like that. And again, I think that's something that, the truth is that most institutions' data are not perfect. There are particular taxonomic and geographic biases, and it's up to the users to make sure that they use the data appropriately. If they're doing research and wanting to publish the results, the peer review process should pick up on any kind of misapplication of the data sets. But again, the idea is we want to get the data out there and used as widely as possible. People are concerned about being scooped on their research. So this is one that's come up quite a lot. So people don't want to publish records of new species that they've discovered because somebody else might kind of come in and scoop them on that and publish the species before they do, or something to that effect. And within the data chapter, the sort of with the discussions that we had with the data working group when we developed this chapter, we came up with a set of embargoes, basically guidelines for doing embargoes on data so that we can protect researchers' interests. There are concerns about security of specimens, and this is a legitimate concern, particularly with paleontology collections, where there's a very high demand for specimens, for high-value specimens, and specimens might actually be stolen if the data are published. And so with the paleontology collections in particular, we've decided we're not going to make them publicly accessible. We'll put them behind some kind of login so that we can validate, only people that are verified bona fide paleontological researchers can have access to those records. But for extant taxa, for the living animals and plants, we want those data to be openly accessible because of the wide range of sort of directly applicable uses that they have for the benefits of society. And then with regards to those taxa, we do obviously have sensitive species where we need to provide some level of protection for the data. So sensitive species are those species where making the data publicly accessible might result in some kind of threat to that species in the wild. A good example are these little succulent plants that you find in the Northern Cape, where the species have got very, very limited geographic ranges. Some of them only occur on one hillside, and there's a very high demand for them internationally. You get people who collect these little succulent plants, and every collector wants to have every species in his collection, and they will go to any lengths to get those specimens. So we know about this being a huge problem in the Northern Cape. There's extensive poaching of plants from the wild, and so we don't want to publish those specimen localities publicly. Again, they would be behind some kind of a login, and only verified researchers or conservation biologists would have access to those data. All right, and then something else that's very important is attribution. Obviously, with all of this open data access, people must still be properly credited for the work that they do and the data that they produce and all of that kind of thing. And this is something that's very well recognized in globally with relation to data sharing, and we're trying to develop this culture of attribution, and encouraging people to properly attribute data sources that they use. So I'm sure some of you might have heard about the Creative Commons and the various Creative Commons licenses that exist, which basically let you put a little stamp on your data set or on your image or whatever it is that you're sharing, and you're basically saying, if you use this, please attribute appropriately to our institution or to me as a researcher, or to whatever whatever the appropriate attribution is. And I think this is something that's really important that people start taking up in the peer review process. So if you are a researcher and you are reviewing people's articles and you see that they are using data from Gbif or from a museum or somewhere else, you should check that they are providing attribution, and just making that sort of a standard requirement for papers that you review. There's also a really nice tool that's become available specifically for biodiversity data. It's called Bionomia. It's this little website. It's down at the bottom right of the screen. And it's an initiative by David Shorthouse in Canada. And it's basically a site that allows you to tie the names of collectors in your databases, so the people who collected specimens, as well as people who identify specimens, and basically tying those names to unique identifiers for those people, so that these systems like Gbif are able to actually see how many specimens a particular person has collected. And people are talking about making this kind of thing a metric that a researcher or a collections staff member would be able to include in their CV. So, indicators of, so like you've got an H index, which shows the impact of your research. People are talking about indexes for other measures of productivity, like collecting specimens and specimen identifications as well. Okay, so then we're talking about data quality. So this is the crux of this of the data chapter, and we came up with these 4 principles that describe data quality. And it's basically that a data set for a collection should be complete, it should be correct, it should be consistent, and it should be comprehensive. And what that means is, what complete means is that we want to have all specimen records in your collection databased and ideally quality checked in some way or another, to make sure that they are that the data in the database actually reflect the data that are on the specimen labels or in the catalog books. We want identifications to be correct and comprehensive. So identifications of museum specimens are a big issue. In some collections, they've measured this. The guys working on Gbif have actually gone and looked. And in some collections, the identifications are up to 80% of the identifications are incorrect, which is very large. And that's obviously problematic for the way that the data are being used. So having correct identifications is one of the things that we're trying to tackle. And I've also said to have comprehensive identifications, and you'll see that on the next slide, what I mean by that. Data consistency basically means the application of standards, that you're using your fields correctly, the data are all recorded correctly, and that the data meet things like formatting standards and that sort of thing. And in terms of having comprehensive data, that's just, I mean, this is very important, and that's talking about having extra information about your data that helps people to use that data. So when people talk about data quality, they talk about something called fitness for use. You know, so are the data fit for a particular use? You can't know that because you don't know what the uses of the data are going to be once the data are published and publicly accessible. So what we aim for instead is to have comprehensive data with as much metadata as possible in the data set, so that people are able to assess fitness for use themselves when they access the data. So this is an example of what I mean by having comprehensive data. And one of the things that we're doing is trying to standardize the collections databases that people are using. So we've got sort of standard templates for the forms and the fields that people are using for specify, as an example. We don't have the same scope for standardization in Brahms, which is the database the herbaria use. But this is the identification form. So over here you've got specify calls identifications determinations. It's the same thing. You can have multiple identifications for a specimen, which means that different people can identify it as different things, and that history will be recorded. But let's have a look at some of the fields that we've included on this form. So the first one is a verbatim identification form. So what we're wanting people to do is to record the identification in the database. Verbatim means exactly as it's written down. So write down the identification exactly as you see it on the specimen label, with all of the information there, before you start to break it up into the individual fields on the identification form. So here we have on the label, we want people to write it Galela, CF, Adams, Yana, and there's a question mark at the end. This is a photo of this particular species, just for those of you who like snails. But that's what we see on the actual specimen label. We can then go and split that up into the various fields on the form. So there's a taxon field where we write the species name. There's a qualifier field where we capture that CF. And there is a confidence field where we've indicated that the confidence is low because of this question mark that we see at the end of this particular identification. The other thing that we've included here is, so the reason that we want to capture verbatim is because there is interpretation. You have to interpret this in order to fill in these other fields. And people sometimes make mistakes in the interpretations. This is something that particularly happens with geographic coordinates. And so we want to be capturing the data. We want that verbatim data so that someone else who's using the data can see if there was perhaps a mistake in the interpretation. Something else that we're trying to move towards or encourage people to do is to start using taxon concepts. So this is something that's becoming that the global community are starting to encourage quite strongly, is to when you use a taxon name in an identification, you must be specific about exactly what we could call it a version of the name, or the concept that name has been used, the group of organisms in the wild that name is being applied to at that particular point in time. So this is because taxonomy changes over time. People have different ideas about what species boundaries are, what taxon boundaries are, all of that kind of thing. And so if you specify, so this is just a note, this is not the author, this is not the taxon name author. It's typically something, the use of the name by another researcher at a later stage. And then you can say, okay, so in this particular case, the taxon is Galela adamsiana, and the person identifying the specimen is saying it's specifically this species according to Herbert 2011. So that leaves, there's absolutely no ambiguity later on when taxonomy changes and all the rest about what this identifier was referring to when they identified this particular specimen. Okay, then we've got the standard things like determined date and the determiner. That's relatively standard. But we're also encouraging people to record identification methods. We've got a really nice pick list of identification methods, and in this case, we can say that the specimen was identified using a DNA sequence, so maybe we barcoded the specimen, and we have the identification references, the det references. And in this case, I can say what the GenBank DNA barcode was that our specimen was matched with when we barcoded it. So we barcoded, we did a blast against BOLD or GenBank, and we got back another specimen, and it says you're closest to this particular thing. So we're recording really comprehensive information here. And we're doing the same with georeferences. So this is, for those who don't know, this is Specify 7. Specify 7 is orange, and this is Specify 6, which is the desktop version, which is gray and blue, but it's basically the same thing. And here we have our Geo-Reference Metadata. So we're recording coordinates. This might be coordinates off a specimen label. It might be coordinates that we're adding later when we're georeferencing. But when we georeference, we have all of these additional fields. We have things like the uncertainty, and so the uncertainty is the distance around the coordinates that covers the area where the specimen was likely collected. Uncertainty units is whether that distance is measured in kilometers or meters. We can say what protocol we use to georeference. We say what sources we use to get the coordinates, and who georeferenced, when they georeferenced. We can have verification remarks. We have remarks about the georeference, and then we have the verification of that georeference. So has somebody else gone and, maybe a supervisor or something like that, gone and checked that the georeference is actually correct. So we're getting really nice comprehensive data in there that other people can use. So where does all of this stuff come from? All of this, these standards that we've included, and the data chapter is based very heavily on Darwin Core. So Darwin Core is an international standard for sharing collections data or specimen data or occurrence data. It's all essentially the same thing. It's been developed through an extensive. I mean, it's been around for a while. And so it really is it's a nice solid standard for sharing, for working with data. And what we can do is we can look at the Darwin Core standard and say, okay, well, how should we be managing data in our databases and in our collections so that the data are compliant with international standards. So it's primarily a set of terms, so terms or data fields with specific names. Each field has got a definition. The definition is really important. So what data gets recorded in that field, and then it also specifies formats. So for example, catalog number. This is a Darwin Core field, catalog number. It's the same thing as what many people call accession numbers in their collections and in the herbarium collections, but they call them barcodes. So this is why I've got tomato, people calling it different things, but it's all actually the same thing. And catalog number is the Darwin Core standard term that refers to this concept of the code or the number that you give your specimen in the collection. Very important is that it's case sensitive and they need to be written out exactly as they're indicated in Darwin Core. So catalog number starting with a lowercase, no space, and then uppercase N is not the same as catalog number where catalog is with a C and there's a space. They're not the same thing. It's very, very specific as to how it's written. And this is so that machines so that computers can read these data sets and aggregate these data sets seamlessly without having to wonder about whether this is the same thing or not. And then also just a nice improvement, a way to be even more specific is if you put what's called a namespace identifier in front of the field. So in this case, we've got DWC with a colon and then the field name. And the DWC is Darwin Core. It's the namespace that identifies where this field is coming from. So if you're looking at this and you go, oh, okay, cool. So I know that's Darwin Core. It's a field called catalog number. I can go and look up the definition for catalog number in Darwin Core and see what should go into that. There are other namespaces that get used. So Dublin Core is one. Ecological metadata language is another. There are others like FOF and various other ones. And so it's really nice when you have a comprehensive data set that's really indicating very clearly what the fields in the data set are actually being used for. So the Darwin Core standard is also evolving over time. So as people use it and they find that they need new fields or they want to have definitions tightened up, you can go and make suggestions for that. It's not done in any kind of willy-nilly way. Darwin Core is a standard, and so they make the process for making changes quite difficult, and so they're difficult and they take a long time. And there's a reason for that, and that we don't want our standards just kind of chopping and changing. So I'm going to show you quickly the Darwin Core website. So I had it there on the slide, dwc.tadwig.org/terms, and you can go and have a look at this website to see what all the Darwin Core terms are. And they're divided on the right-hand side of the page. They're divided into these classes. There are about, I think it's about 250 Darwin Core terms. It's a lot. We don't actually use most of them, but it's nice that they're divided into these individual groups. So for example, if I click on location over here, these are all of the terms, the Darwin Core location terms. And so here's country, there's state, province, county, municipality, locality, verbatim locality. Here's decimal latitude, decimal longitude. These are all fields that we should be relatively familiar with. And if you click one of them, so if we click on locality, it takes us to that term, and here's the definition, which says the specific description of the place. It's got comments, and then it's got examples that show you that what should go into that locality field. So a really, really nice resource for constructing data sets. So what we did in the manual is we developed we identified a core set of fields that we want people to use. Like if you're sharing data or looking at what your forms should look like and specify, these are the fields that we looked at. It's a whole jumble of fields over here. I'm not going to look at them individually, but they are there in the manual for you to have a look at. And what you can also do, if you're creating a new data set, for example, and you want to use some of these fields, you can pick them out of the manual and use them. What you can actually do is you can copy that whole blob of text in the manual and paste it into a CSV file. And then because the terms are comma separated, when you open the CSV file in some kind of data editor, it will give them to you as the column headings, and you can just go and delete any that you don't actually want. So it's a nice thingy there. All right, Darwin Core also specifies data formats for different kinds of data. So dates are the big one. So we've always had problems in the past with how people format dates. And so we use the Darwin Core format, which is actually just the international, it's the ISO standard date format, which is year, month, and day. This is an example. They're separated by dashes, very important. So year, dash, month, dash, and day. If you don't know the day, you just leave it off. So this is a year and a month. It's got ways of indicating date ranges as well. So you indicate a date range with a slash. We separate lists of items with pipes. So these vertical bars over here are pipe characters. So if you've got multiple collectors, for example, you separate the collector names with pipes. Or if you've got multiple collection methods, you separate them with pipes. We don't want white space around strings, no full stops on the ends of strings, and also just not to have any hidden, you get hidden these funny characters that sneak into databases and mess things up, and we want those to be removed. We need to preserve special characters. So this is also something that we often see that gets messed up in data sets. So these characters that have got these accents above them, things like degree symbols, and anything that's not a normal letter, those not a normal letter or a normal number, those get scrambled when data get moved between different data sources. And so one of the principles is that we want to preserve those special characters. We've got a standard format for how collectors and identifiers should be indicated. So it's the surname first with a comma, and then the initials separated by full stops. And then obviously we want people to start using controlled vocabularies. So Tadwig is developing controlled vocabularies. The examples on that Darwin Core page that I showed you often have those controlled vocabularies there that you can use for particular kinds of fields. And then just in terms of the format of data sets, we're also trying to encourage people to move away from using Excel because of when you work with data in Excel, you end up with all kinds of problems. Excel scrambles special characters, and it does all kinds of funny things with dates. The latest version of Excel, the one that you get in Office 365, is a little bit better. It's a little bit more respectful of original data formats. But we want, people should be moving data around in CSV format. So comma separated values, it's basically just a text file. The different fields are separated by commas. It's nice and simple, no fancy anything. And it's less likely to break data sets when you move them. So like, for example, if you do an extract from your database and send it to someone who's requested the data, send it as a CSV file and not as an Excel file. All right, so then looking at data collection in the field, the thing that we're most interested in here is locality data, because that's the primary data that people record in the field when they're collecting specimens. Very often they'll just, they're not recording comprehensive identification data. If they record identification data at all, they might just, if it's a plant, they might just say a family or a genus, or, preliminary identification of some kind. But what is important in the field is the locality data. And one of the really important things here is please record precise locality data, a text description of where you collect the specimens, and the specimen coordinates. So if you've got a GPS or a phone, or however you're recording your coordinates in the field, please record both. What we've seen, a trend we've seen since people have started using GPSs is they don't record the locality data, they just record the coordinates. And then if you get back and there's a mistake in the coordinates, like if you wrote down a number wrong or something like that, or if your GPS was giving you incorrect coordinates, that which GPS devices do sometimes malfunction, you don't have the actual locality data that you can fall back on. So really important that the locality data and the coordinates verify each other. You want them both there to back up the other one in case there's a problem. Also important is to record coordinate metadata. So that's some kind of measure of uncertainty around your coordinates. So what most people are doing is getting out of the car and they record the coordinates there where they get out the car, and then they walk up the hill and they collect their specimens on the top of the hill. So if you can just indicate more or less what that distance was, so the distance from where you recorded the coordinates to where you collected the specimens, so it's you can think of it as a big circle around the point where you stood when you recorded your coordinates, and also to record how you recorded the coordinates and the datum. So most GPSs are set to WGS84. It's the default datum. If you don't know, the datum is actually a descriptor of the shape of the Earth that your coordinate system is using, and different datums have slightly different coordinates for the same point on the surface of the Earth. And if you don't record a datum, you're essentially automatically adding a 500-meter uncertainty to your coordinates. If you don't record the certainty also, and someone wants to geo-reference your coordinates later on, yes, I said it, geo-reference your coordinates, they will have to try and estimate what your uncertainty was. And what we're seeing is we're ending up with like 1 to 2 kilometers uncertainty if people just recorded the coordinates with no extra information. Then in terms of habitat data and weather conditions, these are good things to record as well. And it's a good idea just to try and standardize the way that you do that. I recently bought myself a little handheld weather station, which I use for recording conditions when I'm collecting scorpions at night. And it really adds a huge amount of value to the data that I'm collecting. So that's something that people can look into. And then very important is to, you need to be recording these data somewhere. Collectors typically have a notebook or something like that where they record the data. And ideally, we should be archiving those as well. So notebooks should be going into institution libraries. People are starting to use digital devices like phones to record their data in the field. We don't quite know yet what the best way is for archiving those kinds of data, but it's good to archive that kind of stuff. All right, and then if you're doing georeferencing, please just follow a protocol. That's really important. The NSCF, we've built this really nice georeferencing tool, which our team is using, and it is available to anybody who's wanting to georeference their own data as well. And this enforces the standards for recording comprehensive georeferences. So this form on the right-hand side shows all the georeference information that's being recorded. So there's the uncertainty to the units kilometers. There's the datum, WGS84, who georeferenced it. We include the identifier for the georeferencer as well. That's part of the attribution thing. The date, here's the sources, there's the protocol. So it's a really nice tool for doing rapid, very high quality georeferences. All right, so the next thing that's really important with this data chapter is all of the, well, for all of the chapters in the NSCF manual actually, is that we've got, each chapter has a set of workflows that are required for that particular chapter. For the data chapter, it's a lot. We've got a lot of individual workflows that are associated with data management or management of digital assets inside these collection institutions. And so, so we're looking at things like workflows for recording data in the field, your workflows for data capture and quality assurance or quality control, workflows for managing born digital data, workflows for data backup, et cetera, et cetera. Data publication is a really important one. Processing data requests is another really important one, where it would be good for institutions to have nice, well-resolved workflows that they follow, so that people are getting everything that they need, that there aren't things being left out or mistakes being made, that kind of thing. So this is something that we've been focusing on quite strongly with the data work that myself and my staff have been busy with. And we're wanting people to drop their workflows using workflow diagrams. Now, there's a standard way for drawing workflow diagrams. It's a process diagram, and there's a standard set of notation that you use for doing that if you're doing nice, proper workflow diagrams. And it's not a lot. It's only four items. It's circles, So these circles, which indicate the start and the end point of that particular workflow, the open circle is the start, the closed circle is the end. Very important that every workflow has a starting point and an end point so that it's clearly defined. It's got these diamonds which indicate decision points. So this is like the kind of thing where you say, we're data capturing a specimen, or we're quality controlling a specimen, and we find this kind of mistake. Do we go this way or do we go that way in the workflow? That's what the diamonds are for. So you write inside the diamond what the decision point is, and you have a yes going in one direction and a no going in the other direction. And it tells you which steps to go to. And then these sort of rounded rectangles are the individual steps in a workflow. So these are small, discrete steps. It might be something like extract. If you're doing a workflow for a data request, it might be extract data from the data set, from the database. That's the first step. The next step might be standardize fields to Darwin Core terms, that kind of thing. And by drawing these workflow diagrams, you're able to see where there are inconsistencies within your workflows. For example, this particular branch over here doesn't go anywhere. So when I draw this, I can see, okay, this doesn't actually reach the endpoint. There's something wrong. Maybe there's a step missing, or maybe I just need to draw the arrow to the endpoint. But it gets you to be able to make sure that your workflows are complete and consistent. And often it just with the process of sitting down and drawing your workflows, you start to think, hang on, but if we do it slightly differently, it will go much quicker. It'll be much more effective. So, and much more efficient. So it's a really nice way of thinking about your workflows as well. One extra thing that you can add to workflow diagrams are what these things called swim lanes. So you basically divide your diagram up into these different they're swim lanes like you've got in an Olympic swimming pool, and you have different groups of people or different people. So this is our workflow that we use for data capture within the NSCF. We've got the technicians over here. We've got the data specialist who manages the technicians, the start process here, and then the work moves to the technicians, and then the technicians send the data back to the specialist. There's a decision point here that the data might go back to the technicians, or the data goes out this way and comes to the coordinator for final checks. So really nice to have these. But then so what's important is the workflow diagrams are a tool for developing your workflow, for thinking through the process, formalizing. But what you need to do is write it down in steps, in bullet points. That's your work. The final workflow is a set of points, bullet points that someone can look at. Okay, so that's essentially it. So the next discussion the next session that we're going to have is the discussion session next week. And what I'm going to ask is for the folks who are attending that to please review the data chapter of the manual. Just have a read through it. And note any questions that you might have when you're looking at that chapter, and we'll start off with a little bit of discussion if there are questions that people have about data management in their institutions, and then the bulk of that discussion session will focus on developing workflows. So basically, just playing around with workflow diagrams. We've already run this process with some staff in some of the institutions, and it's actually quite a fun exercise. So we'll just spend some time playing around with workflow tools and developing some workflows. So what I'm going to ask is, before then, if you can just maybe draw your own workflow diagram to start, like think to yourself so this can be any workflow diagram. It can be work related, it can be not work related, it can be related to something else that you're involved in. You might want to draw a workflow diagram for your processes for procuring new computer equipment at your institution. At Sandy, that would look like a spider web or something like that. But you can just use any workflow diagram that you might like. And then let's bring those in when we meet next week, and we can have a look at them. and we'll start playing around with workflows more extensively there as well. All right, so that's it, guys. I see that there were a couple of points that came up. I'm just going to have a look at the Q&A. I see there's no questions in the Q&A, but Zaituna did ask about free platforms one can access for workflow diagrams. So the one that I like is called diagrams.net. I can show you that quickly. So so if you just go to, I'm just going to pause. If you go to diagrams.net, you can download the software there, or you can work with it online. It's up to you, and you can start pulling these things in, and then it lets you draw arrows. If you take the arrow, it lets you draw arrows from one box to the next. You can then write inside the boxes. It's a very, very nice tool. And then if you move the boxes, it looks after the arrows for you at the same time. So that's a nice one to use. All right, so I'm going to stop sharing then. Okay, so any other questions? Any comments? Is there a way to make new Darwin Core identifiers? Question from Cameron. So to have new Darwin Core terms, yes, there is. There is a process. So they now run the change process for Darwin Core on GitHub, and you go and you add a new issue in GitHub, and you say, I want a new term that describes whatever it is, and these are the reasons that we need it, and this is what the definition should be, and this is examples of how we would use it, and this is how people might use it. And then they wait to see if there's sort of verification from other people that that people are interested in using the same field, and then if there is, then it gets added. And it takes, I think it takes about 2 years for a new term to be added in Darwin Core with the review process. I know that the Darwin Core terms for paleontology are a little bit tricky. They're a little bit superficial, but there are other standards that people are developing for geological data, which, to better manage stratigraphy and things like that, which you can use as well. Yes, there's a lot of discussion around paleontology terms in Darwin Core. So Cameron, it might be an idea for you to join the Paleo Happy Hour, which is it's a group of people, mostly Americans, but they discuss exactly these kinds of things, the use of Darwin Core. They meet once every 2 weeks, and they discuss data-related issues for paleontology. It would be really cool if you could join that. I'll send you the link. Remind me if I forget. Okay. Chanel, I will leave the closing of the meeting to you when you want to pull the plug.
Okay, thanks, Ian. Yeah, I think we're done. If you do have any other questions, you can post it on our Facebook page or contact Ian directly. Otherwise, we'll see you at the discussion forum next week. Thanks, everyone.
Yeah. You can get me on ian@nscf.org.za if you want to drop me an email, or in Slack if you've joined the NSCF Slack channel. Yeah, thanks, guys.
Thanks, everyone. Thanks, Ian.